This is our lecture on DNA repair, and our objectives are as follows. Number one, we will learn why DNA repair is necessary. Two, we'll learn what causes alterations in DNA. Three, we'll learn about the different types of alterations in DNA. And number four, we're going to learn on how these alterations are being repaired in the DNA. So the first question that we want to ask is, why is DNA repair necessary? Well, I think it is a rhetorical question because we know that DNA repair is necessary because of DNA damages. And there are a number of things that cause DNA damages. For instance, reactive oxygen species that are formed from normal metabolic pathways. When they're elevated, they can cause problem with the DNA. The chemicals in the foods that we eat can cause alterations in our DNA. Cancer therapeutic agents can also cause alterations in our DNA. And then one that not very many of us think about on a daily basis is that activation of chemicals that are pro-carcinogens. In other words, they are not carcinogenic in their original form, but upon them being taken into our bodies and converted by our metabolic enzymes, they convert to very carcinogenic products. And one example is the benzoapyrene, which is a aromatic compound that is found in tar, coal tar. And then when you take it into our body, maybe through the use of coal tar, breathing it in, or eating things that are cooked on charcoal, and when you take it into our bodies, it is converted to this compound, aromatic compound, which is very carcinogenic. So this is a pro-carcinogen that becomes carcinogenic upon conversion by our metabolic enzymes. What are the types of DNA damages? Well, one of them is, and we're going to outline four of them. Okay? It turns out that if you were to incubate DNA at 37 degrees, that it will go spontaneous alteration. And this is called spontaneous depurination. And due to cleavage of the bond that binds the purine with the deoxyribose sugar and leaving the backbone intact. And this is called abasic, apurinic. And there's an average of 2,000 to 10,000 depurination that occur within a cell in 24 hours. So leaving DNA at 37 degrees unprotected will undergo automatic depurination, where the bases, the nitrogen bases, are being removed, the purines are being removed. The second type of DNA damage is oxidation by reactive oxygen species. And you get formation of 8-hydroxyguanine, and again, hydroxylation of the 8 position of the nitrogen base. This actually has been discovered that it increases with age. The third type of DNA alteration is alkylation of bases by alkylating chemotherapy. And so when you treat a patient with chemotherapy, that chemotherapeutic agent is to interfere with the DNA, and alkylating agent, they alkylate the DNA to prevent it from being replicated in rapidly dividing cells, and so their cells will die. But the problem is that it also has effect on rapidly dividing normal cells, and so alkylation of bases in normal cells can occur, and that's one of the types of DNA damage, and then the one that we're all familiar with, and that is UV radiation to form pyrimidine dimers. Uh, and so the four different types are, again, spontaneous removal of purine bases from the DNA, oxidation, formation of 8-hydroxyguanine, alkylation of bases by chemotherapeutic agents, and formation of pyrimidine dimers by irradiation with ultraviolet light. And so mutations can either be inherited, they can occur during DNA replication, or they can even occur during DNA repair. And so these are the stages where mutations can actually occur in a DNA molecule. Let's talk about some point mutations. Types of point mutations. One of them is transition, and that is when purine is mutated to another purine or pyrimidine mutated to another pyrimidine. Transversion, when purine is mutated to pyrimidine or pyrimidine is altered to purine. Missense, which means that a alteration in one single base will lead to alteration of amino acid. This is a missense mutation. Or nonsense mutation, when a codon is altered to become a termination codon. And so it leads to the formation of a truncated product or a termination of the formation of that product as well, depending on where the stop codon is inserted in the open reading frame. Silent or synonymous mutation is a change in the base, single change or single point mutation that does not lead to any alteration of an amino acid. So not all mutations will lead to, not all alteration in bases can lead to mutations, because some are silent or synonymous. It won't lead to any change in the amino acid product. And as you remember from your basic biology, this is because of the redundancy of the genetic code. Insertion or deletion are single point mutations that are very deleterious, because when you insert or you delete, you cause a frame shift that will either terminate or it will lead to the formation of a product that is different from the product that is intended or that is coded by that open reading frame. There are 2, 3, 4, and 5 nucleotide repeats in the DNA that can also cause alterations. This is common in the human genome. When you have region of repeats, a lot of repeats, this leads to high mutation rate, and it can misalign during replication, leading to insertions and deletions of a number of repeats. And an example of this is the triplet expansion in Huntington disease. There is a repeat that consists of CGG, CGG, and so the number of repeats expand in Huntington disease. And this is, as you know, CGG codes for glutamine. And so expansion of this region will lead to formation of a large polyglutamine region in a protein product. And this is what happens in Huntington's disease. The fate of the alteration. Well, number one, it can be the alteration in the base can either be tolerated, that is, it's ignored, or it can be repaired by mechanisms that are built into our systems, or it can kill the cell and cause the cell to or cause the cell to kill itself, or it can become fixed, and it can result in a mutation that is passed down from generation to generation. So mutations are either tolerated, repaired, or lead to the apoptosis of that cell, or it can be passed on from generation to generation. Now, what are some of the consequences of a compromised repair system? Well, the cell will end up with many mutations, okay? Because as we know, that as the cell continue to grow and replicate, the cell is going to have many, many mutations. Within a 24-hour period, there are a lot of mutations that occur. And those alterations or mutations must be fixed immediately, or they're going to cause problems. And so mutations occur constantly, repair is occurring constantly, it occurs all the time, and those that cannot repair, cannot keep up with the mutation rate, this will lead to senescence or a stoppage of cell growth or maturity, and programmed cell death can occur, or malignancy can actually develop where the cell continues to divide without control. So these are some of the consequences of repair. And so the two things that you can remember is, number one, the cell either dies or the cell becomes immortalized and causes cancer. Sources of mutations. Now, the factors that alter DNA bases in the DNA can either come from within or from without the organism. From within are called endogenous. They occur by oxidative damage due to the reactive oxygen species that are produced from normal metabolic pathways, or, as we looked at it before, depurination of bases, removal of purines and leaving a naked backbone. There are some that come from the outside. These are exogenous, and radiation is an example. Alkylating agents such as chemotherapeutic agents that we talk about, or chemical carcinogens, again we discuss either carcinogens or pro-carcinogens that we discuss. So DNA repair then, needless to say, must occur constantly to prevent alteration. And this is why it pushes the error rate to about 1 in a billion error during replication. Without that repair, it will be worse than 1 in a billion. DNA bases can either be repaired directly by very direct mechanism that can repair them, or they can be repaired indirectly by mechanisms that are stepwise in fashion. So let's begin. Let's discuss the first DNA repair. The first one is called base excision repair. And this is a repair that is abbreviated BER. It repairs modified bases such as methylated bases, deaminated bases, oxidized bases, or even abasic, apurinic, or apyrimidinic mutations. And this consists of a number of enzymes. For instance, glycosylases are enzymes that will recognize the lesion, and they will remove the nitrogen base at the glycosidic bond. This is the bond that connect the nitrogen base with the deoxyribose sugar, and it generates an abasic or apurinic or apyrimidinic site. This is followed by AP endonucleases that cut the phosphodiester bond upstream or 5' adjacent to the apurinic or apyrimidinic site, and that AP endonuclease is followed by AP lyase that cuts the phosphodiester bond 3' to the AP side to remove the sugar. Now the DNA band is completely devoid of that one nucleotide at that region, and so DNA polymerase then comes in and fills in, use the 3' end to fill in the gap, and of course DNA ligase closes that nick, just like it occurs in DNA replication. And this is the mechanism of base excision repair, you have glycosylase, recognizes the lesion, AP endonuclease cleaves, follows by lyase, follows by polymerase, and then ligase to repair that system. And this is just the mechanism. So glycosylases will remove the nitrogen base right here. This is the apurinic or apyrimidinic right here, leaving the naked backbone. And then you have the endonuclease that cleave the phosphodiester bond upstream and then downstream to remove this out. And then DNA polymerases comes in and fills in the gap. Okay, and ligase then connects. The next one is nucleotide excision repair, and this is repair of larger lesions that are more than one single base distortion in the DNA. And this is common to pyrimidine dimers or loops that occur. This repair system cleaves 12 to 13 nucleotides or 27 to 29 nucleotides in eukaryotic. Lesion is recognized by a system of enzymes that consists of 4 enzymes, A, B, C, and D, UVR, A, B, C, and D. And this is the system that is in E. coli. So this occurs by UVRA recognizes the lesion and then recruits UVRB, which is a helicase. Helicase then opens up the DNA at where that lesion occurs. UVRC binds, and then it's the one that cleaves the phosphodiester bond 4 and 8 nucleotides downstream and upstream of the lesion to be repaired. Then UVRD then releases the fragment. DNA polymerase fills in the gap, and of course DNA ligase closes off the nick. This is just the cartoon of the repair, nucleotide excision repair. And so you have A that organizes the lesion and then recruits B to bind. B is the helicase that opens up that region and allows C to access and to cleave upstream and downstream of that lesion right here, upstream and downstream of that lesion. UVRD then releases that fragment. DNA polymerase comes in and fills in the gap, and the DNA ligase comes in and joins the 3' and 5' ends to seal it completely. The next repair mechanism is the transcription coupled repair. And this occurs during transcription because when transcription of a DNA occurs, and if there's a malformation in the DNA, that transcription is going to be stalled. The RNA polymerase is going to be stalled at that lesion. And so in order for it to continue to proceed, DNA repair must occur. And it occurs by an enzyme complex that consists of XPG and CSB. And this, the binding of this complex that consists of XPG and CSB triggers the binding of transcription factor 2H. Again, we're going to cover this when we deal with transcription, but this is a helicase. It is part of the transcription factor complex, but it also serves as a helicase, which opens up the DNA where the lesion is. And this kind of changes the conformation of RNA polymerase II so that it loosens its binding to the DNA where the lesion is. And then the CC1 XPF complex then binds and cleaves at the 5' side of the lesion, and the XPG complex then cleave the strand on the 3' side of the lesion. All right. When that is released, then DNA polymerase fills in the gap, and you know the rest of the story. DNA ligase then closes it off. And so this is just a cartoon right here. So you have that RNA polymerase that is going through, and then it finds a lesion and it stalls. Transcription factor 2H is the helicase that opens up that region where that lesion is, and then the enzyme complex will come in and cleave upstream and downstream, releasing that fragment where that mutation or that lesion occurs, and then DNA polymerase will fill in the gap, and DNA ligase will connect it. The next one is called the mismatch repair, and this occurs when the DNA replication is going on, and then when there is a nucleotide that is put in and it's not really complementary to the base on the strand, that's going to be removed, and this is the process of removal. When the wrong base is inserted, distortion of the DNA helix will occur because it just doesn't fit. And so this is repaired by this mechanism. If this mechanism is defective, it can actually elevate a spontaneous mutation rate to 10 to 1,000 folds. And defect in this mechanism or repair mechanism often lead to colon cancer and other types of cancers. Okay, and this consists of again this enzyme system MUDIS and MUDEL. Okay, MUDIS recognizes the mismatch, and it knows which strand the abnormal base is on, because one is going to be methylated, and that is the old base on the template, and the newly inserted base is not going to be methylated, and this is the one that is recognized. MUDEL and MUDIS bind and recruit MUDH, which nicks the non-methylated strand on both sides of the mismatch. UVRD, which is a helicase, will unwind the DNA. Exonuclease will cleave the strand, and DNA polymerase fills in the gap, and DNA ligase then connects the nick. And this is just a cartoon of it, MUDIS and MUDEL. UVRD, so right here, UVRD will help in releasing the fragment, and DNA polymerase 3 and ligase will come in, fill in the gap, and connecting the nick. Okay, so MUDIS, MUDEL, and MUDH, they recognize the lesion, UVR, and cleave the region with the help of UVRD, and that fragment with the mismatch base is released, and then DNA polymerase and ligase come in and fill in the gap and finish it. Now, there's a repair mechanism called direct demethylation that can occur if you have methylation, abnormal methylation of bases, such as T and G, when they're methylated. These can be recognized by glycosylase and repaired by the base excision repair mechanism. But there are methylases that will actually demethylate that. Okay, and this is called the O6-methylguanine methyltransferase. Okay, MGMT is the abbreviation. This enzyme allows the transfer of the methyl group from cysteine, from the base that is abnormally methylated, to the cysteine on the enzyme. Now, here's what happened. The enzyme, once it accepts that methyl group on cysteine, that enzyme then, that protein is destined for destruction by proteasome. And so you can look at it this way. The repair of one abnormal methylation will destine this enzyme for destruction, even though the repair occurs. And so in that way, we can say that this process is very, very, very expensive. And so this is just a really brief introduction to DNA repair.